Today, we are diving into a world that is completely invisible. It's the universe of microbes that lives on us, in us, and literally all around us. Well, forever. This world has been a massive blind spot in our understanding of biology. But what if we could finally create a map for this hidden territory? Let's take a look at the tool that's making it happen.
Think about that for a second. How do you map a world you can't even see? We're talking about these incredible ecosystems teeming with billions of organisms, and they're all completely invisible to the naked eye. Scientists have been trying to chart this world for a long time, and now we're getting closer than we've ever been before.
So what's the first huge hurdle in mapping this invisible universe? Well, for over a century, the only real way to study a microbe was to grow it in a petri dish. The problem? It turns out that over 99% of microbes on Earth just flat out refuse to grow in a lab. This created this massive gap in our knowledge and led to the birth of a whole new field, one designed to study this unculturable majority.
And that field is called metagenomics. The idea behind it is actually pretty simple, but it's incredibly powerful. Basically, forget trying to grow microbes one by one. Instead, scientists just take a sample from, anywhere. The soil, the ocean, your gut, and they sequence all the DNA in that sample at once. It's kind of like skipping individual portraits and just taking one giant group photo of the entire community's genetic blueprint.
But, this creates a totally new challenge. Once you have all that genetic data, you need something to compare it to. And the issue is, our existing databases are almost entirely filled with the genomes of that tiny fraction of microbes that we can grow in a lab. So just imagine trying to explore the entire world with a map that only shows like a handful of small islands. That is the huge wall that metagenomics has been up against.
So, this brings us to the genetic puzzle itself. Trying to analyze this jumbled up soup of DNA from thousands of different species all mixed together is, it's insanely difficult. It's like trying to assemble a million different jigsaw puzzles that have all been dumped into the same box.
To solve this genetic puzzle, scientists really have two main approaches. The first one, called read mapping, is kind of like looking for puzzle pieces that match the pictures on the few puzzle boxes you already own. It's fast, sure, but you're going to miss every single piece from a puzzle you've never even seen before. The second, much more powerful approach is assembly-based analysis. Now this is like trying to build all the puzzles from scratch without any of the box pictures to guide you. It's the only way to discover brand new things, but as you can imagine, it is unbelievably complex.
And when I say complex, I really mean it. A researcher has to juggle a whole bunch of separate software tools. Each one has to be configured just right, and then trying to get them all to work together, it's a total logistical nightmare. This makes it super difficult to make sure that the results are consistent and reproducible, which, is the absolute cornerstone of good science.
And this is where the hero of our story comes in. Faced with this enormous genetic puzzle and this messy, complicated toolbox, researchers were desperate for a better way. They really needed a single, unified solution that could handle the entire process, from start to finish.
What if you could have just one tool that did everything? A tool that takes that raw, jumbled genetic data from a sample and just automatically draws a detailed map of the microbial community, revealing all the species inside it, even the ones no one has ever seen before.
And that tool is Atlas. It's an open-source software package that basically acts as a complete automated pipeline. It takes a researcher from that raw, messy sequence data all the way to the finish line, fully reconstructed microbial genomes that are already annotated, which is just a fancy way of saying the software has already labeled the genes and figured out what they do.
So how does Atlas solve all those problems we were just talking about? Well, first off, it's user-friendly, which puts this powerful kind of analysis into the hands of way more scientists. Second, it integrates all the best tools into one seamless workflow, so that whole logistical nightmare just goes away. And finally, it's automated and packaged up in a way that it runs the exact same way every single time for every single user. This ensures the results are robust and reproducible, which is absolutely critical. Okay, so how does it actually work? Let's peek inside the black box. Atlas breaks this monumentally complex task down into a really clear, four-step blueprint for building genomes right from scratch. It all happens in 4 steps. First up, quality control. Atlas just cleans up all the raw data, getting rid of any junk. Second, assembly. It starts piecing the short little DNA fragments together into longer strands, which are called contigs. Third, and this is where the real magic happens, is genome binning. Think of this as sorting all those jumbled puzzle pieces by color and shape. Atlas cleverly groups the contigs that belong to the same microbe together, basically reconstructing individual genomes right out of the chaos. And finally, annotation. It goes through each new genome, identifies all the genes, and predicts what they do. So that all sounds great on paper, right? But does it actually work in the real world? To find out, researchers put Atlas to the test. They fed it data from the gut microbiomes of just 8 mice to see what it could dig up.
The results were, well, they were stunning. From just 8 mice, Atlas reconstructed 374 distinct microbial genomes. These are what we call metagenome-assembled genomes, or MAGs for short. And the really amazing part? The vast majority of these were from organisms that had likely never been seen or studied by scientists before, ever. And these 374 new genomes weren't just some minor part of the ecosystem. As you can see here, they accounted for a staggering 75% of all the genetic data in the samples. Just think about that for a second. Without an assembly-based tool like Atlas, 3/4 of this entire microbial world would have stayed completely invisible. So the takeaway here is crystal clear. Atlas works. It successfully shines a powerful light on what scientists call microbial dark matter, that vast, uncultured majority of life that we knew was there but could never properly see or study before now.
And this brings us to the bigger picture. A tool like this isn't just some cool technical achievement. It's a gateway to brand new discoveries that could completely change our understanding of biology, health, and the environment.
You know, as the creators themselves put it, Atlas fills a huge gap by giving scientists a method that is both robust and reproducible. In short, it takes this incredibly difficult and often inconsistent process and makes it standardized and reliable. It's effectively democratizing this really powerful form of science.
And that's really why this all matters. By making this kind of deep metagenomic analysis accessible to way more scientists, Atlas can seriously accelerate the pace of discovery. Whether we're talking about finding new antibiotics, understanding chronic diseases, or tracking the health of our planet's ecosystems, the potential is just immense.
So, for the first time, we have a truly reliable map maker for this vast, invisible universe of microbes. And it leaves us with a really exciting question. Now that we have a better map, what new worlds are we going to find?